And so she's going to be telling us a little bit about the DYRK1A research study that's going on at the University of Washington. Well, she's at the University of Indiana now, but I'll let her do all that talk. So you guys will learn more about that, and then we'll have information for you guys about that later. So we're going to go ahead and get started.
All right. Thank you so much for the introduction. Can folks hear me okay? Okay, wonderful. Well, again, thank you so much for having me. I was hoping to join you all in person, but as they said, COVID had other plans for my weekend. So I'm sorry to be joining you virtually, but I'm also happy that I still have the opportunity to talk to you a little bit about the Tiger study and the work we've been doing at the University of Washington to learn more about DYRK1A. And like folks said, many of you probably know me from my previous role at the University of Washington, where I was a research scientist working on the Tiger study. I've recently switched to a new faculty position at Indiana University, where I'm excited to keep doing research and clinical work with folks with DYRK1A. Let me see, how do I get this slide to go forward? There we go. Okay, so I'll start off by talking a little bit about the history of the Tiger study and what the goals are of the study, kind of overall. Then I'll talk a little bit about genotype and phenotype in DYRK1A, which is really the main goal of the Tiger study, to learn more about that and do a quick overview of both of those. Then I'll end by sharing some of our hot off the presses Tiger study updates. These are from a paper that we're writing currently and are about ready to submit for publication for DYRK1A, and we're excited to have your group be some of the first to hear these findings. And then I'll make sure to leave some time for Q&A at the end. So starting off by talking a little bit about the Tiger study, its history, and its goals. So the Tiger study is located and hosted by the Research in Autism in the Brain Lab, or the RAB lab, at the University of Washington in not so sunny Seattle. And it's located at UW's Center on Human Development and Disability. And we also do some research with the Seattle Children's Autism Center as well. And if you look at that picture there of that kind of drab brick building, we've been fortunate to see many of you there to participate in the Tiger study in the past. And the goal of the RAB Lab is to do research and science to improve the lives of people with neurodevelopmental disorders and their families, with a special focus on rare genetic differences like DYRK1A. So right now, the principal investigators of the Tiger study, these are the folks who are really in charge of the whole study. Emily Newhouse, PhD, is a clinical psychologist and faculty member at the University of Washington. And then Evan Eichler is a geneticist at the University of Washington. We're also fortunate to have many collaborators on this study, including Rachel Earle, Rafe Bernier, Caitlin Hudak at University of Alabama. These are all folks who work on the Tiger study now and in the past. And then we also have a really big team of clinicians, psychologists, research coordinators, geneticists, and we're also fortunate to host a lot of trainees working on our studies too. So undergraduates, medical students, folks who are really excited to learn more about DYRK1A and genetic differences. So Tiger stands for the Investigation of Genetic Exome Research, and our focus is on learning more, like I said, about DYRK1A, as well as other genetic syndromes, rare genetic syndromes associated with autism. So there are a couple different genes that we're really interested in. DYRK1A is really one of our primary genes or syndromes of interest in our study. We've been looking at DYRK1A since the very beginning of the Tiger study several years ago, and are continuing to do so for the foreseeable future. The Tiger study is funded and has been repeatedly refunded by the National Institute of Mental Health, or NIMH. And that's really neat because it's hard not just to get funding for these studies, but also for NIMH to say, yes, we want to keep funding these studies. We think this research is really important. And the primary goal of the Tiger study is to understand how genotype, so our genes and DNA in the body, impact phenotype, so what we see when we interact with our kids or family members with DYRK1A on a day-to-day basis. And the Tiger study takes a genetics first approach where we recruit folks to the study not based on a diagnosis of autism. It's really based on whether they have that genetic difference, so DYRK1A or another genetic difference that we're interested in. And the way we think about kind of genes and things like that in the Tiger study is that complex disorders like DYRK1A really need a complex and multifaceted approach to understanding them. If we just did a few surveys or we just looked at genes, that wouldn't give us the whole picture of our folks with DYRK1A, and we wouldn't really learn as much from that. So we look at both genotype, and what genotype is really our DNA. So if you look at this little picture over there, you can see our DNA is genes that tell the body how to do things. And we do that in a couple ways in the Tiger study. One is when folks participate, if they have access to their child's gene report, which sometimes can be hard to dig up, but if you have access to that, we do ask for a copy of that and take a look. And then when folks participate, our genetics team does whole exome sequencing, where they look at all the genes in the body that make protein. So they look at how the DYRK1A gene is functioning, as well as all the other genes in the body too. Right now, folks are participating in the Tiger study remotely, which I'll talk a little bit about more in a bit and what that looks like. So we're gathering that information through remote saliva collection for the most part, where we send tubes to a family and have a lot of tricks for helping folks collect that saliva from kids who may not be super interested in doing so. And then in phenotype, we're really focused on two big areas. One is the medical complications that go along with DYRK1A. So looking at things like febrile seizures, feeding problems, those things that you all are super familiar with are we really want to help with those too. So we gather a lot of medical information as part of the Tiger study. And then we're also really interested in that clinical phenotype as well. So looking at things like autism or intellectual disability or other developmental differences. And that's really what my talk will focus on more today, is that clinical phenotype in DYRK1A and how we gather that info. So I'll start by giving a quick overview of sort of the genetics or genotype of DYRK1A. And some of this will be very familiar to some of you and might be a review of some things I've talked with you about before, but I do like to make sure to go over it just because I think it's important stuff to go through and can be a little complex. So the DYRK1A gene makes a protein that's called a kinase. And what a kinase is it's an enzyme that speeds up chemical reactions in the body or makes them go faster. And what we've learned so far about the DYRK1A gene and the kinase that it makes is that it's really important for brain development. And that's kind of the number one thing it's involved in. So it's involved in making new brain cells, and it's also involved in fixing connections between brain cells. And we think that that's why when we see folks with DYRK1A, there's such a big impact on kind of brain or neurological functioning and development, is because the DYRK1A gene really has a lot to do with the brain and how the brain grows and develops. We also know that the DYRK1A gene plays a big role in early development. So all the genes in our body are kind of expressed more at different times. They're doing different stuff at different times in our development. And we found that the DYRK1A gene is really doing a lot prenatally and then early in infancy. So we think that's also why we see some developmental differences in DYRK1A pretty early on, is because the DYRK1A gene is expressing really early and playing a big role there. DYRK1A syndrome is caused by loss of gene function, also called underexpression or haploinsufficiency. And what that means is that one copy of the DYRK1A gene isn't working right. It's not making enough kinase. And that, again, is going to impact how our brain cells grow and develop. So again, DYRK1A syndrome is caused when one copy of the DYRK1A gene, because we have two copies of all of our genes, have a change or event, or also called a mutation in the DYRK1A that affects how a gene works. And gene mutations, we actually all have some gene mutations in our body, probably a whole bunch of them. Some gene mutations can have no effect, they can have a tiny effect, or they can have a big effect. They can also be rare or common. So there are some gene changes that are super common. They're observed in maybe 1% to 2% of all people, whereas other ones are rare. In DYRK1A syndrome, the mutations that we see are rare, so they happen in less than 0.5% of the population, and they have a big effect on health and development. So that's, again, why we see kind of these overall differences in DYRK1A, is because they're mutations that are happening in kind of an important gene and are having a big effect on the whole body. The types of mutations or differences that can happen are where a piece of DNA, so if you think of those letters that make up of DNA, a letter might be changed, added, deleted, and that's what's affecting how the protein works. Sometimes folks ask if there are any recurrent mutations that have been identified in DYRK1A, and we haven't found any so far. So most of your kids or family members with DYRK1A, everybody will have a different mutation for the most part. So it'll be a different letter or part of that gene that's affected, but it leads to a similar consequence, which is the protein not working right. So some of the types of mutations or events that might occur. So one is where a letter gets switched, and that's what you can see in that picture on the left. So sometimes if a letter gets switched, for example, it's supposed to be TTC, but it switches to ATC. Sometimes that might not do anything. Sometimes it might cause the gene to stop making a protein, or sometimes it might cause the gene to make a different protein. So missense is where it makes a different protein. Nonsense is where the gene stops making the protein. And those are two types of mutations we see commonly in DYRK1A. Another type is called a frame shift, and that's what you can see over on the right. And that's where a letter gets added or deleted, and it kind of moves over how the letters are read. So it's kind of a reading error where the body reads those letters wrong. And again, that can make it so that the wrong protein's made or that the protein's not made at all. So most mutations that we see in DYRK1A syndrome are de novo. And de novo means that it's a genetic change in the egg or the sperm that happened totally randomly. De novo mutations are not inherited from the mother or the father. So again, they happen totally randomly. And they're not caused by anything that the mother did either before or during pregnancy. We really think that they are just kind of random errors or random differences that happen when a baby gets made. And they happen either during or right after conception, so right away. If your child has a de novo event, the recurrence risk of DYRK1A in future pregnancies is typically less than 1% because it's a totally random thing that happened. But genetic counselors are the best people to talk to about these kinds of questions. And that's also if you have questions for siblings of your family member with DYRK1A or other family members. Genetic counselors are really the ones who can dive into your family's specific genetic event and answer questions like this the best. We do also see some inherited events in DYRK1A. So if a person with a DYRK1A event has a child, their child has a 50% chance of inheriting that DYRK1A genetic difference. And we have seen a few folks in the Tiger study who have an inherited event. We also know that two people in a family, so siblings or parents, can have the exact same event, but look really, really different. And that's one of the things that we're really interested in, and kind of one of the big goals of the study is to figure out why people with DYRK1A can have the same event or an event that does the same thing, but still look really, really different. And what we think that might have to do with is the other genes that we have in our body and how those work, as well as potentially environmental factors that might occur in the womb or early in development. But we need to do a lot more research on this. All right, so that was my whirlwind tour of DYRK1A genotype, and happy to talk about that a little more during the Q&A if folks have questions. And next, what I'd like to do is talk a little bit about how we learn more about the phenotype, so those things we can observe in folks with DYRK1A, and how we're currently gathering that information through participation in the Tiger study. So like I mentioned, during the COVID-19 pandemic, the Tiger study has shifted to having our families participate in Tiger remotely. So for those of you who participated in Tiger in the past, some folks always participated remotely, but we did have a lot of folks who would either fly out to the University of Washington for multi-day visits, or occasionally we were able to come to folks' homes and do home visits there. So initially, we pivoted to remote assessment as part of the Tiger study for safety concerns related to COVID-19. For a long time, it wasn't safe for especially kids with DYRK1A who might be medically complex to travel long distances to get to UW. And we continue to think that that's a priority for a lot of families, that there are some families who aren't able to travel to our events because they're medically complex or because it's just really hard for their kids to travel, which we understand. So safety is one of the reasons why we're continuing remote participation for our medically complex kids with DYRK1A. Another is access. Remote participation in research studies really does enable more families to participate. So again, these might be families where it was hard for them to get on a plane, hard for them to take time off work, where they didn't want to take their kids out of school. We have some more flexibility now to see folks where they have those types of challenges, as well as international families have much increased participation now that we've moved to a remote format. And then finally, there's reduced burden on some of our families. Like I said before, folks would have to take off days off work and come in for multiple weekdays to do some of our study assessments. Now we can schedule those at times that work best for you, often evenings, weekends, or just times that work well for you. And our coordinators are good with working with folks to do that. So there are, of course, some drawbacks to remote participation, mainly that it was really fun to see kids in person, and now we're not doing that as much. But we also think that there are some benefits to that, which is why we're continuing with it for this wave of the Tiger study. So when folks come in for the Tiger study or are participating remotely, we use a lot of different methods to gather information about phenotype. Again, because DYRK1A is a really complex syndrome, and what we do is called deep phenotyping, where we're trying to get as much information as possible. So if you participate in the Tiger study, you will be asked a lot of questions and we'll get a lot of information from you. So there are standardized surveys that we have folks fill out asking about things like behavior and communication and medical problems. We do a lot of interviews with parents and caregivers because you're the expert on your child with DYRK1A, and that's one of the best ways to gather information. And then we're also doing direct telehealth assessments with kids and parents with our clinical team on the Tiger study. And the domains that we look at are really broad. We're interested in cognitive skills, so thinking and problem solving. We're interested in adaptive skills, so those day-to-day functioning skills that our kids and family members use to get through the day and get along with others. Because DYRK1A is associated with autism, we ask a lot of questions about autism and autism features. We ask a lot of questions about language skills, so expressing language and understanding language. We gather information about treatment history because we're really interested in knowing what therapies and services kids have had, which ones work best for them. And then we're also doing some interviews to learn more about psychiatric history. So looking at things like depression or anxiety that kids might experience. So when we do telehealth assessments, we're able to gather a lot of the same information that we could in person because we've really had a lot of practice doing this during the pandemic. So if you have a child who is able to participate in Zoom school or in telehealth sessions, even if it's just for brief periods of time, there's a lot of information we can get. So we can do standardized assessments of cognitive skills, both verbal and nonverbal skills. We have some standardized interactions and diagnostic tools that we can use to look at autism features remotely. So either where they're interacting with a clinician over telehealth or where you're playing and interacting with them and we're observing that. And then we can also do some assessments to look at receptive and expressive language skills over telehealth. So a lot of them are the exact same assessments that we did when folks came in for Tiger in person, but we've adapted them for telehealth. And then I also like to say that I know that some of your family members are not going to do telehealth, and that is completely fine. Because like I said, there's a lot of information that we can gather based on interviews, surveys, observations, where they wouldn't have to sit at a computer. So sometimes folks are worried about that, but that's perfectly fine if kids are not able to do that. Another feature of the Tiger study I like to highlight, because it's a little different than other genetics research studies, is that we ask a lot of questions about parents too. So we might ask you to do some of the same activities that we have your child do to look at problem solving skills or language skills. And that's because one of our unique goals of Tiger is to understand how parent phenotype might contribute to some of those differences in child phenotype. And what that contributes is to our goal of understanding why individuals with DYRK1A have such unique strengths and challenges. And we think that some of all those other genes that they get is probably contributing to that as well. So our genetics team gathers genetic information from parents as well with your consent. And then our clinical team does some assessments as well. And I want to stress that all the information that family members provide is both completely confidential and completely optional. So if there's ever a question you don't want to answer, if there's ever a measure you don't want to do, or if there's one family member who want to participate, but another one really isn't able to, that's all perfectly fine as well. And our coordinators can work out those choices with you. So when you participate in the Tiger study, some of the things that we like to give back to families for their time and participation. One is, in the past, folks have received a clinical report, and we're continuing that. So you'll receive a clinical report with assessments results, recommendations, any applicable diagnoses that we were able to provide. So we are sometimes able to provide first-time diagnoses of things like autism or intellectual disability. And those are signed by a licensed psychologist so that you can use them for things like sharing with school, sharing with doctors, getting services. And that's something that we think is a really important part of the Tiger study because of all the time that you and your family members provide. You'll do a feedback session with one of our research clinicians where we'll talk to you a little bit about your child and about DYRK1A, and you get more opportunity to answer your questions. There's compensation for your time. And then also of course, all the information you provide is going to improve future medical care for people with DYRK1A. That's ultimately our #1 goal. All right, so next what I'll do is I'll get into some of our hot off the presses research updates from the Tiger study and talk a little bit about the work we're doing right now to better understand DYRK1A. So our current research sample, so the group of participants that I'll be talking about today, it consists of 29 individuals with DYRK1A, which to some folks might seem like a small number. To us, we think that's a big number, and we're really happy to have 29 folks with DYRK1A that we can share information on. 27 of those folks participated in in-person evaluations with us prior to the COVID-19 pandemic, and then 2 participated in remote evaluations. We have also, in our current wave of the Tiger study, we've seen 3 additional participants with DYRK1A. We're still finalizing those data, so those won't be included in what I'll talk about today, but they'll of course be included in future analyses. The average age of our participants is 10 years old, but we'll see anyone 4 and up. So we've seen folks ages 4 through 24 through the Tiger study. We're about 50-50 male and female, and we think that's a good representation of what the gender or sex split is in DYRK1A. We haven't seen that more boys or more girls are likely to have DYRK1A. It's pretty evenly distributed. We have seen a lot of white families, so always hoping to get more racial and ethnic diversity in our samples in the future. So I just want to pause and thank you all for contributing to this number and this big sample we have of folks with really rich and detailed information. So next, what I'd like to do, like I said, what I'm going to focus on today is sort of that clinical phenotype. So talking a little bit about what some of the DSM-5 or psychiatric diagnoses are that folks with DYRK1A meet criteria for in our sample, just to start out. So a lot of you probably know that DYRK1A is associated with global developmental delays or intellectual disability. And what an intellectual disability is challenges with both cognitive skills, so that's thinking, learning, problem solving in verbal and nonverbal areas, as well as adaptive skills. So that's areas like communication, daily living skills, social emotional skills, or motor skills. And for that, we're just comparing folks to same age peers. So if you think about comparing your child with DYRK1A to a child who's about the same age and doesn't have DYRK1A, we're taking a look at whether they have challenges in both of those areas compared to those kids. So 24 out of the 27 folks that we've seen, 89%, did meet criteria for intellectual disability, or if under 6, the diagnosis that we provide is global developmental delay. So what this tells us is that most, but not all, individuals with DYRK1A have intellectual disability or global developmental delay. And I like to highlight that because I have seen some things online or some previous research studies that have said that everyone with DYRK1A has an intellectual disability, and that's not true. We do find because DYRK1A has that impact on brain development, it does have an impact on things like learning and problem solving skills, but not every single person we see with DYRK1A will meet those full diagnostic criteria for intellectual disability. When we look at autism, we see that a really similar number of folks, so 23 out of 27 or 85%, meet criteria for autism spectrum disorder. And when we're thinking about a diagnosis of autism spectrum disorder, we're looking at two areas. So one is differences in social communication. So that's things like interacting with others, nonverbal communication, forming relationships, as well as restricted and repetitive behaviors. So that's things like repetitive body movements, repetitive speech, strong interests, sensory differences, and trouble with changes in routine. So what we find again is the same thing, that DYRK1A is really strongly associated with autism, and most, but not all, individuals with DYRK1A have autism. And today what I'll be talking a little bit about is some of the differences that we see in folks with DYRK1A who meet criteria for autism versus don't meet criteria for autism. So the paper I'm going to talk about today is really focused on answering the question of what autism looks like in DYRK1A. Some of you might have heard the saying that if you've met one person with autism, you've met one person with autism. So it's very broad. You can't generalize. And that's the case for DYRK1A as well, that we find that our folks with DYRK1A who meet criteria for autism still have these really specific patterns of strengths and challenges. And we see that in rare genetic disorders all the time, that folks might meet criteria for different disorders, but in a really specific way, where there might be some specific areas they're really good at, specific areas that are really hard for them, specific type of symptom profiles. And that's something that we're really interested in determining more about for DYRK1A, so that clinicians and families can kind of know what to look out for and what might be some good areas for targeting intervention. So for this research study, what we did is we took our sample of 29 folks with DYRK1A and we compared them to two other samples we have of people with autism. So one is kids and teens with autism and average nonverbal cognitive skills, and the other is a group of kids with autism who have below average nonverbal skills. So nonverbal skills more in that intellectual disability or global developmental delay range. And what we did was we took a lot of the measures we have in the entire TIGER study, compared our DYRK1A group to those groups to kind of figure out what's unique about DYRK1A as far as that autism profile is concerned. So one of the areas we look at, if you participate in the TIGER study, you might remember doing a really long 3-hour interview called the Autism Diagnostic Interview Revised. And this is an interview where we ask a lot of questions about really specific symptoms, features, characteristics related to autism. So it's really based on what you tell us about your kids with DYRK1A. And based on parent report from the ADIR, we looked at those specific items and we found areas that are particularly challenging for our individuals with DYRK1A. And this is compared to other people with autism. So one of the areas we found that are really challenging for a lot of our folks with DYRK1A is called social emotional reciprocity, or back and forth. So it's that ability to have kind of a back and forth social interaction with somebody else in a way that feels kind of natural and reciprocal. So for that, we look at things like, if somebody smiles at you, do you smile back? If somebody's having fun, if you're having a really good time, will you turn to your mom and kind of want them to be excited about it too, share your enjoyment with others? And folks with DYRK1A can absolutely do these things. They're just more challenging across the board. So that's an area that we noticed you all telling us that is particularly challenging for your kids, is that domain of social emotional reciprocity. Another area we notice as challenging, or we can also think about it as just a difference, is differences in nonverbal communication. So that's things like some people use a lot of gestures when they talk. I personally use a lot of hand gestures when I talk. Some people do not. And our folks with DYRK1A are more likely to not use a lot of hand gestures when they're talking. Another area might be facial expressions. For some people, it's really easy to read how they're feeling by looking at their face. For our folks with DYRK1A, that might be more challenging, or they might have more of a flat facial expression or more of a neutral facial expression, or some folks you can see they're kind of smiley all the time, so it's a little hard to tell how they're feeling. So we notice them having more challenges in nonverbal communication as well, compared to other folks on the autism spectrum. And then one area I wanted to highlight, because I think it's relevant for a lot of families, is that every single person who we did this interview with told us that their child with DYRK1A has trouble forming friendships with same-age peers. And there are a lot of reasons why that might be the case. I think one is a lot of our folks with DYRK1A have challenges with expressive verbal communication, and that's something that other kids don't always know how to deal with and maybe have a hard time overcoming. But I just did want to highlight that as a potential area for intervention and support, and that if you feel like your child with DYRK1A is having trouble making friendships, that's really the case for other kids with DYRK1A as well, that's a particular area of challenge. When we look at some of those restricted and repetitive behaviors, what we see is a profile where there are some that are happening really often and some that aren't happening as often for our folks with DYRK1A. So our folks with DYRK1A really like to engage in repetitive body movements. So that might be things like hand flapping, arm flapping, head rocking, standing up on toes while moving your body. A lot of you told us that that's something that your kids like to do, and I can definitely agree with that from working with folks with DYRK1A, that that's definitely a self-soothing and calming tool for a lot of your kids and family members. You also reported that your kids do a lot of repetitive use of objects. So that might be doing the same thing with a toy over and over, lining up toys, gathering or collecting toys. When we think about the sensory domain, you told us that sensory seeking is more common than sensory aversions in folks with DYRK1A. So what sensory seeking might look like is, a lot of you have told me in the past that your kids really like water. So going into water, playing with water, being near water. It might also look like interest in smells, in textures, so touching soft things or certain things, in looking at things up close or at an angle. That's something that most parents with DYRK1A, of kids with DYRK1A report to us. On the other hand, the sensory aversions that we see a lot in kids with autism, so that might be things like, having certain textures you won't wear, certain types of clothing you won't wear, smells that really bother you. We definitely see that in DYRK1A, but it's less common than the sensory seeking profile, which we think is interesting. And then our kids with DYRK1A are less likely than other kids with autism to show what are called compulsions or rituals. So that's things like wanting to do things the same way every time, or having a really specific way you have to do something, or that your mom or dad has to do something the same way every time. Again, we do see that in some folks with DYRK1A, but less common than other folks with autism. A couple other findings that we found. So looking at adaptive behavior, our kids with DYRK1A have more challenges with communication and daily living skills than other kids with autism, even those who have kind of a similar nonverbal cognitive profile. And what that tells us is that those are good areas for intervention and support for folks with DYRK1A, and that there may be some other factors leading into difficulties there. For example, medical problems or more significant speech and language problems. When you look at kind of social skills overall, social skills of kids with DYRK1A are really similar to other kids who have autism and intellectual disability. So we would expect to see challenges in those domains, and you see that both in kids with DYRK1A and in other kids with kind of that profile. And then you also told us that while kids with DYRK1A have those restricted or repetitive behaviors, so those repetitive body movements, those sensory seeking, they're not necessarily a problem. So there's something that may be a problem for some families, but it's not necessarily something that's really impairing daily life for your kids. So on some of our measures of repetitive behavior, you reported that your children's repetitive behaviors are less kind of problematic, impairing, concerning than they are for other kids with autism that we see. So that might be, I think that's a strength for our folks with DYRK1A, is that while these repetitive behaviors are really present, they may or may not be causing really significant problems in day-to-day life. So qualitatively, we also took a look at just some of our notes and our observations from when kids came in to see us for the Tiger study. So a lot of your kids have pretty limited speech, so they're using few words or no words when interacting with clinicians. And we also noticed that sometimes those words, those vocalizations have what we call kind of an unusual intonation. So the pitch might be flat, it might be high pitched, it might go up and down a lot. So some of those differences you can hear when talking with somebody or listening to their vocalizations. So some of our folks with DYRK1A who have less speech use more showing and pointing to communicate. Other folks don't really use those communication methods, but some of them do. And one of the areas we noticed across the board when doing kind of activities with folks with DYRK1A is that their response to social situations, so social initiations from a clinician, social interactions, might be unusual or might be a little different than you might otherwise see or expect. We also noticed that our kids with DYRK1A often were really interested in interacting with their parents or familiar folks, but maybe had less interest in social interactions with an examiner. And that makes sense. It's a new person who's less familiar, so maybe a little less interest in interacting there. And we did notice sometimes that when kids with DYRK1A came in to see us, that they had some really strong interests or restricted interests. So I remember talking with a lot of you about your kids' interests in areas like water, elevators, roller coasters, things that move. And that's something that we've noticed is kind of definitely a special interest in the DYRK1A community. And then also noting folks having some minor difficulty with changes in routine or transitioning between activities when they come to see us. So this last part I want to talk about is just findings from a really small sample. So just the few kids we've seen who have DYRK1A and who didn't meet criteria for autism. So when they came in to see us and we didn't provide that diagnosis of autism, why was that? And the main reason is that these kids showed some really notable social strengths. They were really socially motivated, so had a lot of motivation to interact with the clinician, with other folks working on the study, with their parents. Did a lot of what we called shared enjoyment. So if they were having fun with something, maybe looking at you, smiling with you, kind of trying to draw you into it. Frequent social overtures. So again, making a lot of attempts to interact with the clinician or other folks working on the study. These folks tended to have more eye contact than folks who met criteria for autism. But notably, these kids still showed a lot of the same repetitive behaviors as kids who met the full criteria for autism. So they still had those sensory interests, they still had those repetitive body movements. The main difference is really those social strengths that we saw in that small group of kids who didn't meet criteria for autism. So that's one area I want to highlight, is that while those repetitive behaviors are really common across the board in DYRK1A, we do have this subset of kids who don't have some of those social difficulties that you would expect in a diagnosis of autism. And we do not know why that's the case. So that's one of the areas that we're interested in learning about a little more. All right, so what does this tell us? What does this information give us? So when we look at a lot of these things quantitatively, so looking at our surveys and our ADOS scores, so scores on different measures, the autism features in DYRK1A look really similar across the board to our group of kids who have autism and intellectual disability, our group of kids who have autism and those lower nonverbal IQs. But when you drill down, you see that there's a specific pattern of strengths and challenges in DYRK1A. But you really need to look at kind of these individual questions and individual features to find that information. Some ideas for intervention targets based on this. I imagine these are things that a lot of you are probably already working on with your family members. One is developing those social emotional reciprocity skills. So learning how to kind of share your interests with others, engage in a back and forth interaction with others, and maybe doing that as a way to help support friendships. And I do note that I think it's important to figure out if the person with DYRK1A is interested in having friendships. There are some folks on the autism spectrum who really tell you that they don't want to have friendships, that that's not something they're interested in. And that's okay, because they can have rich and fulfilling lives in other ways. But if your child wants to have friendships and is having some difficulties in that area, then targeting that social emotional back and forth, some of those skills might be a way to do that. We've also noticed that our sensory motor interests and behaviors, so again, that's that sensory seeking, those repetitive body movements, repetitive use of objects, those are really clear. We see those largely across the board in folks with DYRK1A, but you're not necessarily telling us that those are areas that are really challenging or impairing or wrong, and that need intervention. And I think that goes along, if folks are familiar with the neurodiversity movement, I think we're learning more about how some of those types of interests can be really good and adaptive for kids and adults and really help them deal with challenging situations. And then one of the areas I want to note is that DYRK1A, so what we see in DYRK1A are these really high rates of autism and intellectual disability. And those are hard to kind of diagnose together. There's been a lot of research recently where researchers have been talking about how it's difficult to determine if folks meet criteria for both of those because a lot of the symptoms overlap. And in our sample with DYRK1A, we've really done the work to see that these kids absolutely do meet criteria for both autism and intellectual disability for the most part. So one of the arguments I want to make in this paper is that if you're a researcher and you're interested in folks with autism and co-occurring ID, that you should really look more at DYRK1A because it might be a good example of what's of a syndrome or of a group of folks who really do meet criteria for both of those and have a specific profile associated with that. So like I mentioned, we have a lot of questions that we still want to answer, and we really need your help to answer those questions. So like I said, one of them is why some folks with DYRK1A have autism or intellectual disability and others don't. We don't know why, and we need more families to learn more about that. And we're also hoping that our genetic information that we're gathering can help us learn about that too. Another question is which tools and measures are most helpful for understanding DYRK1A? When you participate in the Tiger study, we give you a lot of tools, a lot of measures, ask a lot of questions. And one of the things we're hoping to figure out in the future is which of those are really the most helpful and which of those might work the best in a medical setting or a clinical setting for tracking progress or making diagnoses in DYRK1A. Like I mentioned, we're gathering information about treatment history, and that's because we really want to know which treatments families prefer for kids with DYRK1A. So we're asking questions about your experiences with things like speech therapy, occupational therapy, ABA, and that's an area that we plan to publish on in the future and are working on as well as putting that information together. I think a question that a lot of families have and that we have questions about as well is what DYRK1A looks like in adulthood. We're starting to see more young adults with DYRK1A, and I always like to say that we do absolutely think that there are a lot of older adults out there with DYRK1A. They just didn't have access to genetic testing at the time when it was available, because as you know, DYRK1A is something that really has only been identified recently now that our genetic testing has gotten really fancy. So we're hoping to identify more adults with DYRK1A and learn more about that. And then finally, I think the goal of all our research efforts and what we're all trying to do is figure out how we can best support individuals with DYRK1A to have rich, full, and meaningful lives. So have really good lives that really mean a lot to them, where they're happy, they're satisfied, they're well taken care of. And that's ultimately the goal of everything we do in the Tiger study, is to contribute to that. All right. Well, thank you so much for listening to me today virtually. I really appreciate it. I think we do have some time for questions if folks have questions. That's my email address right there. It's ekurtzne@iu.edu. Since I'm not here in person, please feel free to reach out to me afterwards if you have any questions or would just like to chat more about the Tiger study, would like to get connected to folks working on the Tiger study. I'd be happy to facilitate that. And then that website there is the website for the RAB lab that runs the Tiger study out of the University of Washington. And then I'd just like to thank all of my colleagues that I work with on the Tiger study, as well as all of you for your contributions.
Before I hand it here to Adrian, I just want to mention the importance of those research. I think as we learn more, we can really help our kids and adults now in navigate like how they learn better, how we can deal with like the difficult medical conditions. So all of us have to answer tons of medical questionnaires and therapy questionnaires, and I know the burden of that. But I would like to highlight that those type of research are extremely important for the entire community. So yes, it's a lot of time commitment, it's a lot of questionnaires, but it pays off for us, not now, but in the future. So Luna is part of like the new, that just mentioned like the remote access. So Luna is participating on that, and they're extremely flexible in terms of like the virtual portion, right? Luna cannot handle more than 5 or 10 minutes max doing something in front like an iPad. But they were extremely flexible to like, okay, let's do a session. It's going to be 5 minutes now. And we set up another time to keep going. And that worked really well for us. So it is a big time commitment. Thank you. But I wish, like all of us here, all the families can help grow that 29 numbers for the Tiger, and we're gonna have Simon Searchlight talk to us next.
Awesome.
Okay, great. So, like she said, we do have some time for questions. Does anyone have any questions that they would like to ask her? Hopefully I don't disconnect this thing.
So my question is, do you feel like the DYRK mutation is somehow a newer mutation, or do you think that it's always been here and it's just increasing now as population gets larger and larger, or maybe that kids were misdiagnosed with like CP or some other genetic problem? Or do you feel like there could be some environmental changes that could trigger the mutation? Just thinking out loud.
Yeah, that's a really fantastic question. Thank you for asking that. So I think, and again, I should say I'm a psychologist and not a geneticist. But based on the research I've done in DYRK1A, I think it's really kind of the what you said about it not being diagnosed until recently. I think it is something that has always been around because it's something that occurs spontaneously when a baby is made, and it's really something that can happen that has been happening for a really long time. What I think is in the past is exactly what you said, that folks with DYRK1A were misdiagnosed or it was missed with diagnoses like intellectual disability or CP or autism. And that I think there have always been folks around with DYRK1A and that we just didn't know that DYRK1A existed until really recently. And it's only because the genetic sequencing technologies, so like that whole exome sequencing I talk about, that's a thing that they kind of only figured out in the past decade. So we've only had the technology to diagnose DYRK1A within the past decade. To the best of my knowledge, I don't think there have been any environmental factors identified that would increase the likelihood of DYRK1A. Like I said, most DYRK1A is a de novo mutation or event, and we haven't identified anything thus far that increases the likelihood of having a de novo event. I think that's definitely a good research question and something to consider. But right now, to the best of our knowledge, I would say that DYRK1A is something that has always been around, probably at similar rates to now, but we just now have the technology and the knowledge to diagnose it.
Okay, so Eva, I have to kind of walk around to ask people's questions.
Yeah, of course.
Question is asked if you could pause maybe 10 seconds to let me run back over to drop it off to you, or drop the mic off so that way we can have the sound nice of the computer.
Of course.
Does anyone else have any other questions? Oh, yeah, we go.
Hi, Eva, this is Carol Stretfield, Theo's mom. We were involved in the Tiger study early on. I have two unrelated questions. One is, for people who were involved then, do you need their further involvement in any way? Should we be in touch? The other question is about this social emotional reciprocity. A lot of parents who the kids have been diagnosed with autism are sent straight to ABA. And maybe ABA is not the best way to work with that. I think there may be other autism therapies like floor time, like RDI, relationship development intervention, that could help. And do you have any other thoughts about that?
Yeah, those are excellent questions. Thank you. So the first one is whether folks who participated in the Tiger study early on should recontact us and come back. What I would say is that unfortunately, the funding that we have right now for the Tiger study through NIH is not to bring people back. It's to see more people with DYRK1A. So that's not something that the lab has funding for at this time, which we think is a bummer. I think we would love to follow folks with DYRK1A over time. Simons Searchlight does have the resources to follow folks over time because that's a little bit more of the methodology of their study. So I think in the future, there would definitely be interest in bringing back folks who participated in the Tiger study because like you said, it's been several years and things have changed a lot. Right now, what we're funded to do with the Tiger study is bring in more folks with DYRK1A to increase that sample size. So I would say definitely continue to stay in touch with our lab because we'll likely be reaching out in the future. But at this time, the funding is really just to bring in additional families with DYRK1A, unfortunately. And then the second question is about kind of what might be the best sort of autism specific therapies for folks with DYRK1A. And again, that's an area where we're trying to gather that family satisfaction and treatment history data so that we can learn a little bit more about which of those therapies might work best for those areas. I would say that right now, we do typically, when folks come to the Tiger study, we do typically still recommend ABA because that's what's generally recommended for folks with autism and intellectual disability, particularly with building some of those adaptive behavior skills. So things like toileting, dressing, going through daily routines. I think ABA can be really helpful with that. We do always caution parents to try and find a really good ABA provider. There's a lot of variability out there. So it's about kind of interviewing folks, touring centers, making sure that they're, delivering services in a way that's fun for your child and works well for them. But I think you make a really good point that there absolutely may be some other autism therapies out there that would work well for DYRK1A. I think the research hasn't been done on that, which is why I think having family communities like you have is so important so that you all can compare and talk about what worked best for your family members. Does that answer your questions?
Yes, thank you.
I'm not sure if you heard, but she said yes, thank you.
Great.
The important thing, and I know that you understand, is that you're trying to figure out which therapies work. Following people along would be a great idea, but we'll have to look at the Simons. And hopefully one of these days we'll see you again.
Yes, absolutely.
Okay, does anyone else have any other questions? Oh, we have two over here, so give me 1 second. I'm gonna answer. Oh, yeah, if you guys want to come to me, that's helpful.
So you mentioned that DYRK1A is a focus of study, but my question to you is, how does it rank against like other rare syndromes, right? I think, it's that there's a lot to study out there and there's a lot of rare syndromes, and just in the community, how do you feel like DYRK1A ranks in that? Is it a high priority? Is it, is it just another, syndrome out there and we're lucky to get, research money, et cetera?
Yeah, that's a good question. And I'll try to answer it kind of based on my best knowledge. I think DYRK1A continues to be, I think the fact that it's been a gene of interest for studies like Tiger and Simons Searchlight means that it is continuing to be an area of priority for folks who do autism research in particular. So for individuals who are interested in understanding kind of the genetic architecture of autism, what causes autism more broadly, I think DYRK1A is a gene that folks are continue to be interested in looking at. And I think part of that is kind of what we're going to try and say in this paper that comes out is that DYRK1A has really high rates of autism. And that's the case even compared to some of the other genetic events that we look at. So there are some where it might be more like 50% of folks we see have autism or 60% of folks. So DYRK1A, I would say, because of that really strong association with autism, does continue to be a gene of interest. I think what the challenge is for DYRK1A is that it is a really rare gene. So compared to some other gene events that you might have heard of, like Fragile X syndrome or 16p deletions, it's a little rarer. So I think that might be the area of challenge for DYRK1A syndrome going forward as well. But I think researchers like us continue to be very interested and want to make the argument that it's worthy of study and funding going forward. Does that answer your question?
Yeah, it does. Thank you so much. One other question, and it sort of even follows on, is we did NIPT when Luna was born, or before she was born, excuse me, and that gave us chromosomal mapping. And,
Uh-huh.
at the chromosome level, there was nothing out
Right.
of the ordinary that we know of. So my question to you is gene mapping possible through NIPT so people can get an earlier view into what's out there and, possible modifications to DYRK1A?
Yeah. Yeah, that's another good question. And again, this is one where I might be speaking a little out of my element as a psychologist and not a geneticist or genetic counselor. But my understanding is, like you said, a lot of these prenatal testings look at thing at the chromosome level. And what they're looking at are for kind of big chunks of DNA that are either missing or duplicated. So for example, if any of your kids had a chromosomal microarray before they were diagnosed with DYRK1A, that wouldn't find DYRK1A. Because DYRK1A is at the level of the single gene. So in order to find DYRK1A in prenatal testing, my understanding is that you would have to do kind of whole exome sequencing or things that just aren't super commonly done in prenatal testing at this time. So again, speaking a little out of my element, I would say the technology may or may not exist to do that in the future. But right now, with the standard prenatal tests that typically happen, you're right that DYRK1A would not be identified.
Okay, so I think we only have time for one more question, and I saw Anne raising her hand, so I'll end it here and then have a little break before our next speaker.
Hi, thank you so much. This was great. I live in Alabama near Caitlin Budak, and we'd like to sign up for the Tiger study. Where do we all go, those that haven't? How do we begin the process?
Yeah, so you can email me at that email address there, ekurtzne@iu.edu, and I'd be happy to connect you with the current research coordinators. And then also, if you go to that website there's more information about it. And I also bet there are folks at the conference today who can give you contact info as well. But I would say definitely, if you shoot me an email, anyone here, I can connect you to the Tiger study and to researchers at UW. And yes, yeah, Caitlin is in Alabama right now, and she's fantastic.
Thank you. Thank you.
So Anna, someone asked if you would be able to enlarge the screen, so if someone wants to take a picture, that they can. And we'll provide more information for you guys too, in a minute. I don't know if that's possible, Anna, but we just want to say someone had wanted me to say really quick, that we hope that your COVID case is mild, and that we hope you're
Thank you.
really appreciate you taking the time to do this for us, even though you're home, not feeling well. We really appreciate you, and it helps us in our community. And so if we can give a round of applause for Eva.
Thank you so much. Yes, and it was definitely on the milder end of COVID cases. I just know that a lot of your kids are medically fragile, and I didn't want to get anybody sick. So thanks for letting me present remotely.
Yes, thank you. We appreciate that. So we're gonna take just a 2-3 minute break. We have some water, we have some snacks. If you guys need to check in on your kiddos, that'll give us time to get our next presentation ready.